Methods. We extracted the frog sciatic nerve and exposed it to ringer solution at 15, 25, 37, and 60 degrees Celsius, respectively. Upon exposing the nerve to 60 degrees Celsius ringer solution, it ceased producing action potentials. We will discuss what's going on here physiologically at the end. At each temperature, we tested for threshold and recruitment, conduction velocity, and refractory period.
Before we dive into our results, we need to establish the basics of action potentials. We are modeling the membrane of the neuron with a castle wall, which is lined with voltage-gated sodium and potassium channels. Orange swordsmen inside the membrane are potassium ions. Blue archers outside the membrane are sodium ions. An action potential is propagated by the movement of ions across the membrane of a neuron. At rest, the membrane potential is set by sodium-potassium ATPase, which uses energy from ATP to release 3 sodium ions and take in 2 potassium ions, such that the inside of the cell is negatively charged relative to the outside at negative 70 millivolts. Upon receiving a stimulus above threshold, voltage-gated sodium channels open, allowing sodium ions to move down their concentration gradient into the cell. This influx of positive ions gives rise to the depolarization phase of the action potential, where the potential of the inside of the membrane becomes increasingly positive relative to the outside. After some time, the voltage-gated sodium channels inactivate via pore blockage, as shown by the guards, which stops the flow of sodium into the cell. The depolarization of the cell activates potassium channels, which allows potassium to flow down its concentration gradient out of the cell. This efflux of positive ions represents the repolarization phase of an action potential. The inside of the cell becomes less positive relative to the outside. When the neuron becomes repolarized enough, the inactivated sodium channels deactivate, which allows the sodium channels to become depolarized later. Repolarization also gives rise to a period of hyperpolarization, where the membrane potential becomes more negative than resting potential. The membrane then returns to resting potential via sodium-potassium ATPase. Next, we need to explain threshold and recruitment regimes. The threshold of the sciatic nerve is determined by the lowest stimulus amplitude required to elicit a cap. The first neurons to respond to a stimulus are the A-alpha motor neurons due to their large diameter and high degree of myelination, which both contribute to a lower threshold potential. Thus, whenever threshold is reached, the cap is attributed to the firing of these neuron fibers. A-beta and A-gamma neurons are recruited next, requiring higher stimulus potentials than the A-alpha motor neurons. The last of the neuron fibers to be recruited are of the A-delta variety, which are the smallest and the least myelinated of the 4 types. As such, they require the highest stimulus potential to fire, and they set the maximum cap amplitude. Total cap amplitude increases until all available neurons are recruited, which is signified by achieving max cap.
The asterisks in this figure indicate the threshold potentials. Under cold conditions, which is demonstrated by the blue line, the larger stimulus potential is required to elicit a cap. As the temperature warms to room temperature, the required stimulus decreases, as shown in the gray line. Finally, under warm temperature exposure, the required stimulus potential generating a cap is at its lowest, as shown by the red line. What causes the change in threshold as a result of temperature? An increase in threshold depends on the conductance of the ion channels. With colder temperatures, the conductance of ions decrease, and ions flow through channels at lower speeds. In the case of sodium, the lower temperature causes sodium channel conductance to decrease, which affects its response to a stimulus in opening the gate. This increases threshold potential because a higher stimulus potential is needed to overcome the reduced conductance and force open the sodium channels to stimulate an action potential. Following this logic, at higher temperatures, the conductance increases and less membrane voltage is needed to open the sodium channels. This results in a lower threshold voltage at higher temperatures. Finally, going back to the figure, you can see that there is a trend in increased maximum cap as temperature increases. This is because at a given stimulus potential, a certain number of nerve fibers will contribute to the cap by firing an action potential. During warm temperatures, more fibers can contribute to the cap at a given stimulus due to their increased excitability and conductance. In contrast, cold temperatures will result in a decrease in the max cap because of diminished conductance capabilities in some of the neurons.
The conduction velocity when we treated the sciatic nerve at room temperature was 62.5 meters per second. The conduction velocity is the velocity at which an action potential propagates down the length of a neuron. Out of the neuron fiber types, A-alpha neurons are the fastest conducting fiber type because they have large diameters and high myelination that both contribute to high conduction velocity. A larger diameter of neuron increases conduction velocity because less sodium will encounter resistance from the membrane and will therefore travel faster. Increased myelination contributes to conduction velocity because myelin insulates a section of the neuron and prevents ion leakage via ion channels. This induces saltatory conduction and allows ions in an action potential to travel longer distances between nodes of Ranvier and propagate without being slowed down by ion channels. The conduction velocity for the sciatic nerve when treated with cold ringers was 38.46 meters per second. When the nerve is exposed to cold temperatures, the voltage-gated sodium channels that are responsible for eliciting depolarization open at a slower rate. This results in a longer delay between the introduction of a stimulus and the opening of the voltage-gated sodium channels, which means that depolarization leading to an action potential is also delayed and slows down the propagation of action potentials down an axon. This is thus reflected in a slower conduction velocity at lower temperatures. The conduction velocity when the sciatic nerve is treated with warm ringers is 45.45 meters per second. As the exposure temperature of the nerve is increased, voltage-gated sodium channels are able to open faster, allowing for an earlier flow of ions, causing depolarization when compared to colder temperatures, and the speed at which an action potential propagates through the membrane also increases. However, at 37 degrees Celsius, temperature is increased to the point where myelin can be denatured by heat. Increased myelination due to denaturation contributes to decreased conduction velocity, so heat's demyelination decreases the conduction velocity.
Now on to refractory period. At room temperature, the refractory period was 0.9 milliseconds. In terms of refractory period, the slower conductance of the voltage-gated sodium channels causes the absolute refractory period to be reached at greater interstimulus intervals. This means that as the time between two stimulus pulses decreases, more and more of the voltage-gated sodium channels remain in their inactivated state. The time between two stimulus pulses at which a second cap is not present represents the absolute refractory period of all fibers in the nerve, because all of the voltage-gated sodium channels are still in their inactivated state when the second stimulus is introduced. Thus, another action potential cannot be generated. For this experiment, a stimulus of 500 millivolts is assumed to be strong enough to generate action potentials in neuron fibers that are in the relative refractory period. A refractory period where an action potential is more difficult to generate due to some sodium channels still being inactive and a hyperpolarized membrane potential. Thus, the refractory period here is defined by the delay between two stimulus pulses that is incapable of generating a second cap, indicating that all neuron fibers in the nerve are in an absolute refractory state. We can see that shorter intervals of stimulus pulses and the amplitude of the second caps decrease with decreasing time intervals. This is due to the fact that as time interval between stimulus pulses becomes shorter, some neuron fibers begin entering their absolute refractory periods, thus decreasing the amount of fibers recruited to help generate a cap, leading to a decreased second cap. This goes on until all fibers in the nerve reach their absolute refractory periods and no second cap is recorded. Now, in consideration of the effects of cold temperature on refractory period, the refractory period for the nerve when exposed to cold ringer solution was 2 milliseconds. At cold temperatures, deactivation and inactivation of sodium channels are prolonged and therefore create a longer duration of the action potential and longer sodium flow into the cell. The longer the neuron takes to inactivate the sodium channels, the longer it will take to remove inactivation from the sodium channels via deactivation. So, When the nerve is exposed to cold temperatures, the period of time in which the voltage-gated sodium channels are in an inactivated state is prolonged. Deactivation of the sodium channels, which marks the end of absolute refractory period, happens later. Finally, at warm temperatures, sodium channels are activated and inactivated quicker. This leads to a shorter absolute refractory period. As seen in the data, there is a large increase in slope in cap ratios for the warm treatment. This is because at high temperature, almost all the neurons' absolute refractory period have been shortened. There are less neurons with long absolute refractory periods. There is less variability of absolute refractory period times, and the neurons will all have absolute refractory periods at around the same short time. The cap ratio of cap 2 over cap 1 will therefore reflect a large increase in slope because each nerve fiber enters absolute refractory period around the same time interval as one another. In a relatively short amount of time, an increasing amount of nerve fibers enter absolute refractory period simultaneously rather than gradually.
Now that the rest of the results have been covered, we can discuss what happened when the nerve was subjected to 60 degrees Ringer solution. Under extreme heat or heat shock, proteins on the membrane of the cell denature, leaving holes in the membrane called nucleation pores. This increases ion permeability such that sodium and potassium can flow freely across the membrane. The free flow of ions eliminates the concentration gradients that serve as the driving forces of action potential generation and propagation. Furthermore, the concentration gradient cannot be restored because the proteinaceous ion channels have denatured. Hence, the sciatic nerve can no longer produce caps at this temperature.